So you see my first slide?
Yes.
Okay, very good. So I can continue talking. I just see my screen. And if there are any problems, please let me know, and then I will try to fix them on my side. But for the moment, this is the opening slide of my presentation. Is that okay?
Yeah.
Okay, great. So today I would like to talk about epitope-based vaccines, meaning that there are a number of modalities of vaccines that we're all familiar with. And in order for us to appreciate specifically what I'm referring to, I would like to give you just a very brief overview of vaccine history. Now, well over 200 years ago, I'm sure you're all familiar with the fact that Edward Jenner, a physician in Gloucestershire, England, was confronted with having to deal with patients that are ill with smallpox. And he noticed that he saw that milkmaids tend not to get infected with the human disease. And so he looked into this more seriously, and to make a long story short, he realized that milkmaids are often infected by a cousin virus, the bovine, the cow smallpox virus, and he figured out that if he inoculates people with the cowpox virus, this affords protection against the human life-threatening disease, smallpox. So this was the very, very first incident of a vaccination procedure in which live cowpox was used to protect against human smallpox. And in fact, the word cow in Latin is vacca. And so the term vaccine is derived from Jenner's first experiments, and this was in 1796. Now, the next milestone in the development of vaccines is, of course, Louis Pasteur. And Louis Pasteur in France in 1885 used an attenuated version of human rabies virus. He and his colleague, Emile Roux, they developed a method by which they could slowly desiccate the spinal cords of infected rabbits. And from that, he was able to inoculate people. In 1885 was the first case with these live attenuated rabies virus, and that also proved to be very, very effective. So we have live vaccines, and we have live attenuated, and ultimately, in 1955, Jonas Salk, in trying to deal with polio, developed methods in which he could use John Ender's protocols for growing polio virus, picorna, but he also then realized that if he treats the virus with formaldehyde under certain conditions, he can isolate a killed, in quotes, virus vaccine, an inactivated virus which cannot infect nor replicate, but it is quite effective in eliciting protective immunity. And therefore, this was the classical case of a formalin inactivated killed viral vaccine. And so we have here, as I said, live, attenuated, and killed. Now, another example of an attenuated is Albert Sabin's polio vaccine, in which he realized that if he grows his polio virus for many, many generations under suboptimal conditions in different cells, in monkey cells, he's able to generate an attenuated version which provides protection against the disease, poliomyelitis, but it also replicates in the gut and prevents the development of infection in the gastrointestinal system, which is very, very important for polio. And finally, I would like to mention Maurice Hilleman. Maurice is probably the king of vaccines. He is developed so many vaccines working in Mert. Unfortunately, he passed away a number of years ago. But to his credit, he was also the first to develop a subunit vaccine, that is to say, purified protein, the spike protein, the surface antigen of hepatitis B, along with other vaccines that he developed. But it serves as an example of not using the virus at all, but rather just a purified subunit spike protein. And so we see a progression that we've used live virus as engender and through attenuated and killed to ultimately subunit vaccines. Now, the work that we have been doing has taken this one step further. And although we don't have examples of successful epitope-based vaccines, we are convinced that this does have promise and should be beneficial. And so I'd like to share with you some of our work in which we've been working on developing the vaccine modality, which is based primarily on a critical neutralizing epitope of the viral spike protein. And the example that I would like to start with is SARS coronavirus. But as you notice here, we're looking at the year 2002. And historically, this is rather interesting because these young girls are wearing masks. And I remember when I saw this picture in one of the newspapers, I found it striking. Never could I have imagined that some 20 years later, the whole world would be wearing masks for a cousin virus of SARS coronavirus. So this is the SARS coronavirus 1. The total epidemic of SARS coronavirus, which you see here, is a total of about 8,000 people worldwide, where today we're looking at over 200 million people infected with COVID-19, SARS-CoV-2. So these are very similar in their general genetic composition. However, they're very different in their capability of actually being transmissible in humans. And the critical component that I would like to emphasize, and I hope you can see my cursor here surrounding the spike protein, and that is the component of the membrane of the virus, the membrane coat envelope of the virus that interacts with its corresponding receptor, ACE2. And here's a crystal structure of just part of the spike protein. This is referred to, the gray backbone, as you can see here, is the receptor binding domain. It's about 200 amino acids, 193 if I'm not mistaken. And this structure was shown to interact with ACE2, the viral receptor, and it interacts with an extended alpha helix here in red of about 40, 50 amino acids, which interact with these two anti-parallel beta strands of the RBD. Now, all attempts to try to truncate the RBD and make a smaller version of the receptor binding site had failed when people had discovered this interaction. And the reason is if you look, here is, and I got ahead of myself, excuse me, but if this is ACE2, we do know that neutralizing monoclonal antibodies, in this case ADR, which was developed by Wayne Marasco and Jianhua Xui, the CDR loops bind, as you can see, exactly to the same surface. And so the question is would it be possible in some fashion to produce a recombinant version of just the receptor binding motif? And so that's indicated here in red. And all attempts to do that had failed. Now, maybe this is due to the fact that we want to understand better the constraints and the supportive factors within the core of the RBD. And as you can see, there are 4 green beta strands, and there's a central beta strand, which serves as a linchpin. And these 5 strands hold the core together. And any attempt to truncate the core, the whole structure falls apart. So a number of years ago, way back in 2004, we proposed, would it be possible to forego and remove the core, here's the core, and these beta strands, and just try to express this single structure going from residue 432 to 486. And this would be, would it be possible to reconstitute a functional RBM, receptor binding motif? Now, of course, the criterion for success would be that we can express this. That is not a problem using recombinant DNA, but we require that it folds correctly so it can be recognized either by the receptor or by the neutralizing antibody, ADR, that was developed by Wayne Marasco. Now, surprisingly to us, when we expressed 432 to 486, and this is research done by Dr. Natalia Freund, this structure, although it was expressed, had no physiological binding activity at all. And this was curious for us because we felt that there's the sequence and all the information is there. Could it fold on its own? And so it does not bind either ACE2 or the neutralizing monoclonal antibody. Now, when we looked closer at the RBM, the red structure here, and the core, we realized that there is an anchor loop, this that I'm highlighting in blue. And this anchor loop actually forms a number of hydrogen bonds, which are blinking now, with the core. So we realize that whereas this loop does not contribute to receptor recognition or recognition by a neutralizing antibody, it seems critical in the structure of the core that it tacks the RBM, the red structure, in the correct position and is held in position by the loop and these hydrogen bonds. So we made the decision to see maybe if we're expressing the RBM with the loop, it doesn't fold correctly. Could we remove the loop, which creates a situation where we have 2 discontinuous strands of the RBM, 432 to 443. We remove the loop here, and then we go from 460 to 486. So these 2 beta strands, which I'm highlighting, are critical and very important for binding the receptor or the neutralizing antibody, but we need some mechanism to hold them in place in the proper conformation. So the question is how do you bridge this gap and make one continuous structure from 432 to 486? This gap is 7 angstroms long. So here's the sequence, and what we're saying is we would like to remove the 16 amino acids of the anchor loop, which we have done, and if this is the structure, how does one link from the leucine 443 to 460 phenylalanine, or as you can see here, a bridge here? And so what we did is we took our phage display system and we expressed the one segment and then the second segment, and between the leucine and the phenylalanine, we introduced a linker of libraries, or a library of linkers that connected these 2 residues, 443 and 460. We didn't have any capacity to anticipate or model what would be the correct linker. And so here we use combinatorial approach. We basically synthesized a whole bunch of linkers. Initially, we tried to guess. So this is the intact loop that I mentioned previously. It does not bind, neither the antibodies or the receptor. If we simply connect it without a linker, it doesn't bind. If we use 1, 2, 3, or 4 glycine residues, assuming that they're flexible, they too do not bind. And if we actually introduce all 20 amino acids, NNK is a codon that codes for all possible amino acids, 1 or all 400, 20 times 20 amino acid linkers, none of them worked. However, we then went to libraries that contained 3 and 4 random linkers, and what we immediately discovered was here are a bunch of linkers that we see here, a lot of them, GEM, TEP, and so on. If we look to see what the composition of those linkers are, a logo shows that there's prevalence, for example, of a glycine in position 1 and almost exclusively a glutamic acid in position 2, and then variation. And if we do the same for libraries of 4 amino acids, once again, we are very successful in isolating linkers that reconstitute the physiological structure of the receptor binding domain or motif. And here you can see a graph which shows you a histogram in which, first of all, we monitored the actual expression of the RBM by phage display. So this is telling us that in each one of these linkers, there is expression. And then we went on to test, do these linkers reconstitute a physiological structure which recognizes the neutralizing antibody ADR. And as you can see, the vector does not, and the RBM, which is the full length, or the loopless, are expressed well, but they do not bind. But those with the correct linkers, 3 amino acids and 4 amino acids, express well and bind quite well as well. So we were very pleased with that. The question is do these reconstituted receptor binding motifs of SARS coronavirus 1, do they also bind? Are they recognized by the receptor? And the answer is yes. Not all of them, but as you can see, for example, GEM generates a construct which is easily bound by ACE2, as well as these others, which are quite relevant and certainly robust binding. So we were very pleased, and we felt that we've developed a new method. If I summarize briefly, we can say that if you identify the receptor binding motif, you might want to remove those areas that do not contribute to binding and may actually interfere in assuming a physiological and correct conformation. Then you want to produce a conformer library, a library of multiple recombinant combinatorial linkers, and then you can screen that phage display library of potential reconstituted receptor binding motifs against neutralizing antibodies or the receptor, and then you can select by fishing out and pulling down those constructs that have, of this vast collection of combinatorial linkers, the correct linkers that enable the reconstituted epitope to form a physiologically relevant structure, which is recognized. Now, all of this was very interesting, and we thought we had a template, or at least a prototype, for a SARS coronavirus vaccine. But as you all know, by 2003, nobody could care less because SARS petered out and disappeared. But as you can see, in 2012, if this is SARS coronavirus, another beta coronavirus, MERS coronavirus, emerged. And so we decided to try to implement the reconstitution protocol with MERS. And initially, we decided to construct a library and test to see if it would allow the reconstitution of the RBM. So if you look at the two structures, here is SARS, here on the left, and on the right you see the structure for the MERS RBD. Now, there are a lot of similarities. You can see we have the 5 beta strands that stabilize the cores. And then you have the RBM here in yellow or gold. They both have. And then you have the anchor loops in blue. You can also notice that the structure in MERS is much more challenging. It has 4 strands here, as opposed to 2, which is simpler. And there's more complexity at the head of the molecule. And so simply removing from 514 to 527 the loop and cutting at 493 to 564, the notion was is if we made a library, could we find something that binds the receptor for MERS? It's a different receptor. It's not ACE2, it's DPP4. And I'm telling you that all of our efforts in trying to make a combinatorial library for the MERS RBM failed in binding DPP4, CD26. It simply doesn't bind. So here I needed a special solution, my special forces, somebody who would be diligent and creative and able to produce and solve the problem. And here I got import from India, Lakshmi Navasayu Palapati, my special forces. Lakshmi did an amazing, amazing job. And he created the library. And as you can see, after creating multiple libraries, it was very complicated. And he was extremely successful. And we knew that it didn't bind the receptor, but we were fortunate to receive from Mikhail Dimitrov a neutralizing monoclonal antibody that binds to the RBM of MERS coronavirus. And this is one of the original pieces of data that Lakshmi provided in showing that it is possible to reconstitute a MERS RBM using a combinatorial linker library. In Lakshmi's case, he used 3 to 7 combinatorial amino acids. And as you can see, there are quite a few positive strands. And here, the idea that Lakshmi proposed was if we cannot show that the receptor binds, maybe we should use a panel, a collection of different monoclonal antibodies. And here you can see a bunch of them, M336 and 337. And then we also received some monoclonals from Barney Graham at the NIH. And what this diagram illustrates, if here the blue is the anchor loop that we have removed, if we look at what residues from 493 to 565 are actually in contact with the antibodies, these have all been co-crystallized, or with DPP4, you can see that by putting together a panel of monoclonals, we are able to get information on the conformations that are required to provide functional contact residues with this panel of antibodies, even though we know that DPP4 does not bind. So here's a picture. First, here on the left, we see a crystal structure of the RBM in red, the anchor loop in blue, and the core. And for each monoclonal antibody, M336 and M338 and 511, you can see that we've highlighted the position of the critical contact residues required for antibody recognition. And the notion is if we can get reconstituted RBM that cross-reacts with 5, 6, or 7 monoclonals, this would give us the confidence that we have been able to reconstitute a physiologically relevant RBM for MERS. And so, as you can see in this slide, and this is a summary slide, showing 1, 2, 3, 4, 5 different clones. And here are the linkers of those clones. And you can see that they bind the monoclonal antibodies quite well. You can see it here in the histogram. And you can see a general simplistic study or summary. And you can see, for example, E5 or E9, they actually bind all of the monoclonal antibodies. They're very, very cross-reactive. And so we are excited about this, and we believe that we've been able to demonstrate here effectively and very efficiently a reconstituted RBM for MERS coronavirus. Now, to put things into perspective, MERS is not very transmissible in humans. However, it is still a major threat as per the World Health Organization. This virus is transmitted via an intermediate vector, camels, and every year there are about 100 to 200 cases of MERS even today, and the fatality rate of MERS is 30%. If you think about it, CoV-2, the fatality rate is probably percent, maybe 2%, in some areas much less. And for SARS coronavirus 1, it never reached 10%. MERS coronavirus fatality rate is in the area of 30, 32%. And every year, there are 100 to 200 cases. And so it does not transmit well, and it's usually spillover from camels. But we are very concerned about the prospects that if it continues to mutate and emerge, it could develop a better capacity to infect humans, and therefore we are continuing and currently working on this as well. Now, it suffices to say, if we look at the genetic diversity of corona, and we saw SARS and MERS, it wouldn't be reasonable not to mention SARS coronavirus 2. And so you can see that SARS coronavirus and the genetic diversity in the phylogenetic tree is very, very close to SARS-CoV-1. And the main difference is that SARS-CoV-2 has emerged and mutated to be extremely transmissible and contagious. And so the whole world today is confronted with the coronavirus CoV-2, COVID-19 pandemic. And so obviously we are working diligently, and Lakshmi is contributing much to try to reconstitute the RBM of SARS-CoV-2. We already have some leads, and as I want to move on to the next example, it's too early to describe our results in CoV-2. Now, another example, because I'm trying to give you the sense of the capacity and the ability to reconstitute an epitope and then use that as a vaccine, a very serious problem, as you well know in India as well, is dengue virus and dengue fever. This is a flavivirus. It's not a coronavirus. It's also RNA. And the real problem with dengue is the fact that there are 4 serotypes. So people who are infected with dengue serotype 1 mount an effective neutralizing antibody response. However, if they get infected with dengue serotype 2, the antibodies are sometimes generated against serotype 1, the original infection, are ineffective in neutralizing the other serotypes, and more importantly and severe is that they also lead to antibody-dependent enhancement. And therefore, dengue was chosen as a good example to see if one could generate a epitope-based vaccine that would be very effective in neutralizing the serotype and not eliciting ADE. And so here is work done by Chen Piller in my lab. And in fact, we are focusing here and making considerable progress. As you can see, here's the polyprotein of the dengue virus, and the E protein is the envelope. And in the envelope, there are a number of highly well-defined neutralizing epitopes. And you can see here is the crystal structure of the monomer of the envelope protein of dengue, and it's divided into 3 domains. This is the D1 domain, which bridges D2 and D3. Now, the orientation of this highly conformational and complex structure is even more so when you realize that the second monomer of the dimeric structure is juxtaposed in opposite direction. So here's D3, and here's the D3 of the second subunit. D3 is known to contain and harbor a very, very potent neutralizing epitope. It's not an epitope that binds directly with the receptor. The receptor actually binds in an area somewhere around here. But it has been shown that one can develop very, very potent neutralizing antibodies that bind D3. So we decided to evaluate if we could use our methodology of combinatorial linkers and reconstitute a D3 epitope-based vaccine for dengue. And so here's the molecular structure, the crystal structure of D3. And as you can see, it's highly convoluted and complex. And we decided to trim, as I proposed previously, those areas that might interfere with the reconstitution. So for that, we snipped and cut here and there the structure, which led to this structure here. And then we needed to be able to link this area with a linker. We also added linkers at the N-terminus area domain and the C-terminus aspect, but I'm not showing it here. But what you can see is that the concept is the same. So we took the complete D3, we cut it in a number of places, introduced combinatorial linkers, and then screened against 2 neutralizing antibodies, 1A, 1D, and 4E, 5A. And here I'd like to mention our collaborators, Sachdev Sidhu, Dev Sidhu, which you may know. He is also of Indian history and descent. He's working in Toronto, is a brilliant scientist. And Fred Fallouz, who works with them. They are our collaborators in working on dengue. And as you can see, we were able, Chen was able to isolate a number of constructs after screening thousands and thousands of colonies. And she shows that here we have 5 that bind the antibody very well, 1A, and another 2 that bind 4E. But in contrast to what Lakshmi showed, here we do not see constructs that seem to cross-react, bind more than one antibody. And our philosophy was, our protocol said that in order to reconstitute a functional, physiologically relevant epitope, we need to show that it can bind at least 2 monoclonals. So here, actually what Chen had done is she did a reciprocal experiment. She took all of the clones that were isolated with antibody, let's say 1A, and used them to screen for E, and vice versa. And she did this numerous times, constantly enriching for those select clones that actually bind and cross-react effectively both of the antibodies. And as you can see here, we have 4 clones that cross-react very well. They bind nicely, 1A, which is a weaker antibody, but they bind well. And they bind, of course, 4E very well, as compared to other clones that bind 4E or 1A, but not both. So, once again, we were able to reconstitute. And with that, I would like to say thank you. And, of course, I would be very happy to entertain any questions you may have. Thank you very much. and I will stop sharing, and I would be delighted to answer any questions you may have.
Thank you, Professor Kasumi, for a wonderful talk. And we are very privileged to have you here in the conference. Now I request the audience to ask you questions.
Professor John.
Yes.
Thank you. Thank you. Wonderful work. We enjoyed. And I have a small question here.
Sure.
You said combinatorial linkers in SARS virus, antibodies development, monoclonal antibodies development.
Yes.
You showed that they have very good efficiency in binding and as well as cross-reactivity.
Yes.
Are they also showing cross-binding efficiency on these variants, different variants we are noting on SARS virus, alpha, beta, delta variants now?
Okay, so you're asking specifically about COVID-2.
Yes, COVID-2.
And of course, that is an important question. The reality is that for the moment, we have a number of lead molecules that cross-react with two neutralizing monoclonal antibodies. We are continuing to screen, and we know that the construct at least is able to recognize the first of the mutations that was described, the asparagine 501 to tyrosine, which was typical of the UK and South Africa. Now, as you know, the South African construct mutation includes 484 and 417. 484 is in the midst of our RBM, but we have not tested that as yet. We're still trying to accumulate more cross-reactivity. And 417 is not included in the RBM. If you look at the structure of the RBD of COVID-2, 417 contributes a lysine residue that interacts with ACE2, but it does not necessarily, it's not included in our construct. And so I can't tell you if this would be a problem. Now, the Delta and the Delta Plus are interesting because if you look at Delta, the signature mutations there are the residue 452 and 478. And these are included in our RBM. And of course, we will address this specifically as we're developing candidate vaccines. 417 once again pops up as the difference between Delta and Delta Plus. And so, as I mentioned, 417 isn't in our construct. Now, I would like to elaborate a bit, if I have the time, and that is to explain why I believe using an epitope-based vaccine is so important. As we know, when you're using full-length spike, which is the immunogen for almost all of the vaccines, if you look at AstraZeneca or J&J or Gamaleya, they're using adeno-based vaccines that are expressing full-length spike in a DNA presentation. And then Moderna and Pfizer, obviously, are using the messenger RNA, again, for the full-length spike. There has been evidence that the NTD, the N-terminal domain, is relevant, but we all, I think, would agree that a formidable, major neutralizing surface of the spike still remains the RBD. Now, there has been some mention of potential ADE in coronavirus, and I also believe that if it would be possible to reconstitute the neutralizing surface, I believe that this would be very effective in generating a highly cross-reactive neutralizing response because the RBM must maintain its ability to interact with ACE2. So this is a special site in the spike which cannot afford major mutation and variation, because massive mutation in the RBM surface would be a penalty and detrimental to the virus. It would lose its ability to bind the receptor. So in my mind, all of the mutations we are seeing, they have absolutely nothing to do with evasion of the immune system or the vaccine, but rather they are very slight modifications in the periphery of the critical binding site so as to better adapt the virus to bind to the human receptor. And if that's the case, if we can reconstitute the receptor binding motif, that will ensure that whatever antibodies we produce should occlude and prevent virus attachment to its target cell. So I think this is a very valid approach, and I'm hoping that we'll be able to, possibly next time, but have data to show that it can not only be done, but it can elicit broadly cross-reactive neutralizing antibodies that can tolerate the presence of 452 and 478 and 484, because these are very sporadic here and there modifications that I think are attuning the site to better interact with the human receptor, and therefore they're becoming so much more transmissible.
Thank you, Professor. One more question from participants. Can I ask on behalf of them?
I'm here. Happy to answer whatever I can.
Yeah, participant asking question: can we predict peptide MHC scores provide evidence to establish a protein's potential as a vaccine?
So the question of prediction is very, very interesting. And of course, there are a number of different methodologies that are being developed with molecular dynamics and docking and so on. I'm old-fashioned. So, we also develop computational predictive algorithms in another fashion. I'm not overly convinced that the field has matured sufficiently that through first principles, looking at a structure, we can predict what would constitute ultimately a pristine neutralizing surface. I think when we look at co-crystals, or if you want, also of course NMR structures or EM structures of cryo-EM and so on, all of these are very, very much biophysically based in looking at the interaction between, let's say, a surface protein of the virus and its corresponding receptor. And that's telling us what are the critical residues and the critical surfaces. I have not been able to even predict what would constitute an effective linker. And so there again, being old-fashioned, my attitude was remove what we don't need and introduce a vast collection, comprehensive library of combinatorial linkers. It's easier for me to generate 10 million linkers as opposed to think about the correct one. And then I allow the biology to, using affinity selection of neutralizing antibodies, to pull out that reconstituted RBM that seems to have assumed a recognizable physiological structure. So my attitude has been exploit combinatorial chemistry and recombinant DNA and neutralizing antibodies. These are substances that I can work with in the lab. And as you see, Lakshmi's done a wonderful job.
Thank you. Thank you very much. Any other questions from the audience?
If And for me, there is also another question from the other audience. Can I say
Sure.
what you said? The message reads, is actually from Mr. Ravi Lata. My sir, thanks. I have one specific question and curiosity on the mutants in RBD domain. Like, how it is increasing the infectivity rate and binding with S2?
Can you repeat the question again? I understand we're talking about CoV-2.
Yes, sir. It was about COVID-2 genes. So it says like how it is increasing the infectivity rate and binding with S2, the RBD domain.
So you're asking me, if I understand correctly, can I explain or shed any light as to why these mutations that are appearing actually have preferred infectivity, and why at the molecular level do I believe that they're better adapting? Now, that's a very good question, and a question which is very relevant. And if we actually understood the mechanisms, it might also give us some method, possibly, to predict the next variations. However, what I have noticed, I believe that CoV-2 is the product of spillover from bats and then possibly pangolins to humans. And of course, the virus in the wild, in nature, was most adapted to infect the bat ACE2. And only when it spills over into the human population are the initial spillover events interacting with human ACE2 with some level of mediocrity. They're not optimized as yet. And so the more and more cases and transmissions and the numbers of replications in the individual and then in society are giving the virus the opportunity to test here and there slight modifications, and of course a selective advantage to those that acquire more efficient transmission. Now this is the general mechanism. Your question asks, do we understand what, let's say in Delta, 452 and 478 or 484, why are they necessarily increasing and improving the transmissibility? Now, I don't know this for a fact, but I'll share with you what I think. If you look at these mutations specifically, some of them are a transposition or an exchange of a negative charge of a glutamic for an arginine or a lysine. So in my mind, and I'm not sure that this is pure speculation, but I'm happy to share it with you, I believe that what we are seeing is that the surface of the RBM of the original virus, CoV-2, benefits if we introduce a few positive charges. And I generally see, in looking at the mutations, this seems to be the case. Then if you look at the flip side, how this interacts with ACE2, you can imagine that these positive charges, which are the product of mutation, may create an electrostatic field which draws ACE2 more efficiently to the surface to bind, because the surface of ACE2 tends to be electrostatically negative. And so this is very simplistic and possibly very naive on my part. But I haven't heard anybody else who is actually trying to address what your question is. And the way I envision it is that I think that the select mutations tend to increase the positive electrostatic charge of the RBM, which creates a more efficient electrostatic field which can draw in ACE2, which its binding surface, that helix from residue 20 to 55, tends to contain more negative residues. And therefore, it could be something as simple as improving the electrostatic charge interaction between the two molecules. I don't think it has anything to do with evasion of immunity. And in fact, unfortunately, you know as well as I do that when Delta appeared in India initially, the vaccination coverage in India was not high. And so it would be silly to argue that Delta emerged due to vaccination pressure. I don't think that's the case. You seem to be muted. I don't hear you.
Yeah, thank you. Thank you. Thank you, Professor Joe. One more question, if time permits, can I ask?
Yes, of course.
This is from audience. Is there any successful commercially available Epitope vaccine is available?
There's a very
movement.
Yeah, it's a very quick answer. No.
No. All right.
Now, I do want to slightly elaborate.
Right.
There are, as you know, quite a few subunit vaccines.
Oh.
And these are very, very effective. And even if you look at COVID-2, Novavax and Sanofi are developing full-length spike
protein.
We also know that there have been attempts to use the RBD, just the 200 residues of the carboxyterminus of spike, which contains the RBM, as a vaccine. I believe this is a new technology, and therefore we will have to illustrate the advantage of epitope reconstitution. And that is one of the reasons why we're looking at dengue, because we know that dengue vaccines have been problematic due to the ADE. And I believe that dengue would be a good example. And we're also working on COVID-2 because we are all witness to the mutations and the variations. And if we can convince that the reconstituted receptor binding motif affords protection and greater robust ability to neutralize the ever-emerging variants, then a market will be created. However, my whole career has been dedicated to doing the science. I'll let the biotechnologists deal with the market.
Thank you. Thank you, Professor. Any other question?
Yes, Doctor, this is Suresh. I am having a question regarding dengue, Professor Deshwani.
I'm sorry, I didn't get that.
No, Professor, I will ask you. So, you are discussing about dengue.
Yes.
Dengue vaccines, and already one of the vaccines is there. It is not approved by FDA. But my question is, you are talking about the epitope base. Yes, how epitope base is going to answer ADE, antibody dependent enhancement?
Thank you for the question. There are two possibilities. ADE in dengue is perceived, as far as the mechanism is concerned, that when you are exposed or you are vaccinated against serotype 1, you generate antibodies. Some of them are neutralizing for serotype 1, but you also generate antibodies which are non-neutralizing and potentially cross-reactive for serotype 2, 3, and 4. And therefore, if you're subsequently infected with 2, 3, or 4, the anti-serotype 1, which is non-neutralizing, may allow infectivity of immune cells, macrophages, and others via forming an immunocomplex which is non-neutralizing with immature, otherwise non-infectious virus via FC receptor, which introduces them into the cell, and then you can start the chain of events for infection. So that's the mechanism that I believe is rather reasonable for ADE. Now your question is, if I had an epitope-based vaccine, why would it circumvent and afford better protection? And the answer is the following. ADE rests on the fact that you generate non-neutralizing antibodies that can cross-react with immature virus. Otherwise, you don't have a mechanism to introduce via FC receptor. And so if we limit the immunization protocol to only the neutralizing surface, then you do not have the non-neutralizing antibodies which mediate ADE. The second issue, however, is will the neutralizing anti-epitope cross-react with other serotypes? So let's assume I have a reconstituted serotype 1, and that's what I showed you. Will it cross-react with serotype 2, 3, and 4? And so the two monoclonal antibodies that we selected, 4E and 1A, are not only neutralizing, but those are the best cross-neutralizing antibodies that exist. That's why we looked at domain 3, because domain 3 actually generates cross-reactive surfaces. However, if we find that the reconstituted epitope elicits very strain-specific, subtype-specific response, then we will reconstitute the same for serotype 2, 3, and 4, and use a heterologous combinatory multivalent vaccine that will satisfy protection against serotype 1, 2, 3, and 4. That's what we anticipate.
Thank you.
You're welcome.
Any question?
Sir, I have a query. Can I?
Okay.
So that was an informative presentation, and my query is whether it is possible to identify genes that are responding to vaccine through a docking study. So that's quite simple. Yeah.
To be honest, I don't know. My focus has always been looking at antibody interactions with viral proteins. And of course, I've done a lot of work in expressing viral gene, but I don't know necessarily of any vaccine modality that focuses and targets viral genes. I simply do not know.
Okay. Thank you so much. Thank you.
Thank you.
So if there is no further question, I think we will thank Professor Dasoni for a wonderful interaction.
Thank you very much.
And we are very grateful to